My name is Amitavo Ganguli. Welcome to my lightning talk entitled Mining the Plant Reactome Database for the Evolutionary History of Metabolic Pathways. This work is being performed under the supervision of Dr. Sushma Naithani of the Department of Botany and Plant Pathology. Today I'm going to briefly introduce the Plant Reactome Database, discuss programmatic access to the data, and talk about how we've used the data to research the evolution of metabolic pathways. Here's the Plant Reactome Database website. We can go to the pathway browser, drill down into the metabolic pathways on the left, and then examine individual reactions. The information in the Reactome database comes from many different sources, but the standardized representation of the data allows us to query the information using automated tools. In this way, we can gain new insights that weren't imagined by the original researchers. In this study, we use the Reactome database to research the evolution of metabolic pathways. For each metabolic pathway in the database, we find the reactions that are part of the pathway, find the proteins that participate in those reactions and the genes that code for them, and finally we find the orthologs of these genes across all the species in the database. If these orthologs exist across the species in a clade, we conclude that the pathway probably existed in the common ancestor. Note that this sort of analysis is only possible because data from different studies has been standardized and made accessible to automated tools. Here's the data once it's been extracted. Note that the metabolic pathways on the left mirror the information in the web interface. There are reactions, and beneath each reaction are the proteins that are involved in that reaction. Along the top, we have different species in the database, and the main body of the spreadsheet contains the orthologous genes for each species and reaction. Here we see a heat map that demonstrates the result of the analysis. Along the left are the database IDs for the metabolic pathways. Species are along the bottom. The colors represent how well the pathway is conserved in each species. Note that some pathways, visible as horizontal yellow lines, are well preserved among many related species, and some others appear to be common across all the species in the database. I'd like to acknowledge the invaluable help that I've received from my mentor, Dr. Naithani, and the funding from the URSA Engage program and Oregon State University.